Okay, our final speaker is Mike Seganish. He has been working at, or he worked at Merck Sharp and Dome for 17 years in the discovery chemistry group, and over the past 6 years has focused on building capabilities in ADCs and targeted delivery at the South San Francisco site. His efforts have focused on novel ADC targets, novel payloads, as well as novel formats, including peptide drug conjugates. And he will tell us about his talk in navigating undesired payload release toxicity in ADCs. Mike, thank you so much.
Great. Thanks for the kind introduction and for the invitation to come and speak today. So going last is a benefit and a curse. It's definitely a curse because I'm standing between everyone and Friday happy hour. But it's a benefit because there's been a lot of great talks today that have laid a lot of the foundation for some of the things that I wanted to talk about. And the main thing I want to focus on is basically the nemesis for ADCs, and that's the toxicity that's inherent with the payloads that we're conjugating and then dosing into patients. And so I wanted to talk through a couple of ways to think about this, some possible solutions that people have looked at to try to resolve it, and just kind of lead with that it is still an unanswered question as to how we can solve this. There's probably multiple ways to address toxicity. So at this point in the day, I don't need to go through a lot of introduction here, but the one thing that I want to briefly touch on are the ways that ADCs and payloads can actually get into cells, because this is going to be important to put into context some of the issues with toxicity. And so shown on the right here are several ways highlighting these mechanisms of action. So the first of which is the one that is usually the one that we think about for ADCs. That's the target-mediated delivery and uptake into cells and then release a payload there. And so this is what we want to have happen in tumor cells. But the drug and the ADC also have lots of ways to get into cells in different ways. One being a non-target mediated uptake here, where you can get uptake of the intact ADC into cells that don't express the tumor antigen. Likewise, you can have ADCs that fall apart and release the drug over time. That free drug can then permeate into cells and have undesired toxicity that way. But then also, too, you actually have your target-mediated activity or independent activity that can result in catabolism of the drug, release of the drug into circulation. Now you have free drug that can also then permeate into cells and tissues beyond the original cells that had taken this molecule up. So one of the things I wanted to go back and revisit is, again, a piece of data that folks have seen multiple times today, and that's the comparison between TDXD and TDM1. And we've had several discussions today around this efficacy, the really compelling efficacy of Enhertu here, and some of the rationale for why that's true. But one of the things I really wanted to call out here, particularly based on my background as a small molecule medicinal chemist, is recognizing that for both of these molecules here, you're leveraging the exact same antibody. And what you've done is switch the linker and payload and made a significantly better drug that gives dramatically improved efficacy in patients. And so from a design perspective, I think what that teaches us is that we can optimize that linker and payload and have a significant effect on both the ADC activity as well as the potential tolerability. But it's not all good news, right? So these ADCs, even the best ADCs, have a significant amount of AEs associated with them. I just highlighted one paper here where they were actually looking at a doublet or even triplet combination of drugs to try to minimize the nausea and vomiting that's associated with Enhertu. One of the other things that Pooja actually just touched on as well is thinking about the future and where we want to go with ADCs. Right? Almost all chemotherapy drugs are actually given in combination to drive maximum efficacy in patients. And we really want to be able to do the same thing with these ADCs. But if we have a really significant tox burden, that's going to make it very challenging to do so. And so trying to get a handle on this toxicity for the ADCs is going to become important for that. So what I'd like to do is just take a step back for a second here and give just a general overview of some of the tox that has been run into with ADCs. And what was actually done, so a meta-analysis was done over almost 170 clinical trials, looking at over 22,000 patients. And what they saw was that in over 90% of the patients, you saw treatment-related adverse effects that were related to the drug. And of those, about 46% were grade 3, so very serious AEs associated with these drugs. And if you group these AEs or these toxicities into two different buckets, what you have are what are called the off-target, off-tumor toxicities, and then the on-target, off-tumor toxicities. And so this off-target, off-tumor toxicity is actually the predominant toxicity that's seen with most of the ADCs. And what is effectively happening here is that you're getting uptake of the ADC or the free payload into tissues and organs that don't express the target, and then leading to tox. And so a couple of examples of this are the neutropenia and the neuropathy that you see with many ADCs, particularly auristatin ADCs. The other bucket of tox here are the on-target, off-tumor tox. So this, again, is related to the ADC getting to tissues that are normal tissues, but these tissues actually express the tumor antigen that you're targeting for the tumor. And so I'm actually not going to go into a lot of detail around these types of toxicities. You can alleviate a lot of these toxicities with the proper choice of the target, but also potentially pairing with the proper payload. Selecting a payload, for example, that normal tissue is not really going to respond to. But just to highlight one example here, the skin rash that's observed with Nectin-4 targeting ADCs that have MMAE as the payload is an example of this on-target, off-tumor toxicity. So what I wanted to do is actually take a step back now and look at some of the early ADC work and try to put it into context within chemotherapy, particularly some of the work that was done with Trodelvy. Again, shown here on the left, a molecule that's been talked about a couple of times today, and actually compare it to Camtastar or irinotecan, which is the actual chemotherapy prodrug that's used to deliver SN38, which is also the same payload that is used in Trodelvy. And so I'm not going to go through all the PK details here, but what I really wanted to draw your attention to is the amount of free SN38 that's generated from Trodelvy, and then what's generated from dosing patients with irinotecan. And so if you actually look on a molar basis at the Cmax level of SN38, it's about 0.3 micromolar. If you compare that to irinotecan, it's about 0.14 micromolar. So actually very similar Cmax levels of free payload, both with your ADC as well as your traditional chemotherapy. But also too, if you actually look at the exposure on a daily basis, so on a 24-hour basis, uh-oh. It's okay, don't. No? Okay. If you actually look at the exposure on a daily basis, again, comparing SN38 from Trodelvy to Camtastar, they're actually very similar. And so what you're seeing here is actually a very similar exposure for the free drug, both with the targeted ADC and the non-targeted chemotherapy. And so what you would expect, and in fact is what you see, is a very similar toxicity profile between these two molecules, particularly when it comes to neutropenia and incidence of diarrhea. And so thinking about this molecule here, a lot of this is probably coming from the fact that we know Trodelvy is a very unstable ADC. And so this actually leads into a really thought-provoking paper that was published a couple of years ago now by Colombo and Rich from Zymeworks, where they were reinvestigating the original hypothesis behind ADCs. And what that hypothesis was is that the intent of ADCs was to take a drug that's a fairly potent molecule where the minimally efficacious dose and the maximum tolerated dose are very close together. And by making this into an ADC or a targeted delivery, you can dramatically increase that window. And preclinically, you actually see this fairly well, particularly in rodent models. But in practice, when you actually get to the clinic, this starts to fall apart. And when they did their analysis, what they actually found was that the maximum tolerated dose of the free drug is actually very similar to the maximum tolerated dose corrected for the recommended phase 2 dose of the ADC, and leading to a very similar concentration of overall payload that's dosed to the patient. And so in this case here, the real differentiator for the ADC versus the chemotherapy actually comes down to the response. And so for ADCs, you see a dramatically better response in patients compared to the chemotherapy itself. And going back to the example on the previous slide, comparing Trodelvy in triple-negative breast cancer to irinotecan, you can see the response rates are dramatically different. So Trodelvy works much better than irinotecan in these patients. And so the question is, if we can actually make a much more stable ADC with more stable linker payloads, can we actually start to shift this paradigm so that we still maintain that efficacy and maybe can tamp down some of the tolerability concerns? And so one of the molecules I wanted to compare to is actually this molecule on the bottom, SKB 264, also known as sacituzumab TMT. This is a molecule that was discovered by Kelun Biopharma, but then licensed by and now developed by Merck. And so the inspiration of this molecule actually came from Trodelvy, the structure of which is shown here on the top. And I want to point out two key differences between these molecules. The first of which is the payload itself. So SN38 is, of course, the payload for Trodelvy. For sac-TMT, the molecule is this belotecan derivative. If you actually look in vitro, the in vitro potencies between these molecules are actually fairly similar across multiple cell lines. What's interesting here is that the release mechanism for Trodelvy is this hydrolyzable carbamate motif. That motif is the same payload release mechanism in sac-TMT. The main difference between these molecules is actually in the conjugation. So while Trodelvy uses this unstable maleimide conjugation, for sac-TMT, this cysteine arylation chemistry is leveraged to make what is effectively an irreversible conjugation to the antibody, tamping down on the ability for deconjugation to happen with this ADC. And what you can see preclinically, so this is an in vivo efficacy model here on the right, when both Trodelvy and sac-TMT are dosed at the same dose level, you see very little activity for Trodelvy and a really nice efficacy curve for sac-TMT. And it's thought that part of the rationale for this improved efficacy actually has to do with the stability. And so in this graph down here on the bottom, we're looking at the percent theoretical release of payload. So 100% would be all of the payload coming off of the ADC. And you can see the curve for Trodelvy here. At about 24 hours, actually all the payload has come off of the ADC. Whereas for sac-TMT, you do have a significant amount of payload that's still coming off the ADC, but it's dramatically lower than Trodelvy. The question is, though, what does this actually translate to in the clinic? And so this is a snapshot of a clinical trial where both of these molecules were tested not head-to-head, but in the same non-small cell lung cancer patient population. And if you look at the dosing regimens, they're actually a little bit different based on the PK properties of these molecules. Trodelvy was dosed once a week at 10 mpk. Sac-TMT is 5 mpk once every 2 weeks. But you can see for the response rates, again, sac-TMT is almost double the response that you see with Trodelvy. But really interestingly, you actually see the same rates of one of the DLTs in this study, which was neutropenia. And so what we're able to show here is that a more stable linker was able to improve the response in patients for these molecules, but you get a very similar profile when it comes to toxicity. And so the question is, can you actually take this a step further, right? So for sac-TMT, we're still using that hydrolyzable payload release mechanism, but what if you actually went with something that should be much more stable, particularly in plasma? And one of the examples of this has to do with engineered cysteine conjugation, which has been mentioned a couple of times before. But in this example here, I'm exclusively going to focus on the Thiomab platform that Genentech had pioneered. And in this example here, they built two molecules. They're anti-MUC16 antibodies. One of them uses their Thiomab drug conjugate at a DAR of about 1.6. The other one is just a traditional stochastic conjugate, giving you a DAR of about 3.1. And both of these are using the exact same linker payload. It's the vedotin linker payload that was pioneered by Seagen. And so at a high level, if you take a look at how these molecules behave preclinically, again, on this xenograft model on the left here, both ADCs are dosed with single dose at the same dose level. And you can see the Thiomab drug conjugate gives you better tumor regression as opposed to the ADC. That's probably explained in part by the better PK profile for the Thiomab drug conjugate, where you get much better exposure, lower clearance of the Thiomab as opposed to the ADC itself. But what was also interesting was they had some safety data that they have published associated with these molecules. And they actually did, in this case here, a cynoneutropenia study where they were looking at the depletion of neutrophils in vivo. And they compared head-to-head their ADC and the thiomab drug conjugate. And so they did two different measurements here. The first one here, so this is the MMAE dose that's given here. Remember, these ADCs have a different DAR. The thiomab drug conjugate is half of the DAR of the traditional ADC. And so you actually have to give double the dose of the thiomab conjugate to get a similar MMAE concentration. But what you could see is after a single dose, you saw very little impact of the thiomab drug conjugate on the ADC, on the level of neutrophils, compared to the ADC itself, where you saw significant depletion. And so again, some promising data showing that you get improved efficacy, improved PK, and maybe a hint of improved safety here. And so Genentech actually took this example further and took and tested both of these molecules in the clinic. And so on the left here is the stochastic DAR4 conjugate, and on the right here is the thiomab DAR2 conjugate. And so an interesting point here are the doses that were used. So the recommended phase 2 dose was 2.4 mpk for the stochastic conjugate versus 5.2 for the thiomab conjugate. But you can see some of the data here, again, aligns with what was seen preclinically. You have higher clearance for the interchain as opposed to the thiomab. You have a better response for the thiomab compared to the interchain conjugate. You're starting to also see some AEs at a similar rate between the two molecules. But what was actually really interesting and potentially surprising at the time was they saw a new AE that came out of this study here, a significant amount of ocular toxicity with this ADC. And so while they were able to get improved efficacy with this molecule, now they're starting to introduce an additional toxicity here that had not been seen before with these MUC16 ADCs. One of the things that is an interesting observation may be coming to people's mind now, particularly after Greg Thurber's talk, is going back to the differences in doses here, right? So they were able to dose almost twice as much of the ADC on the right, which is giving you about an equimolar concentration of MMAE. And so part of this improvement in response here may actually be coming from better tumor penetration due to that higher dose. Okay, so I don't want to imply that this ocular toxicity is a significant problem only associated with these thiomab conjugates. One other quick example here, this molecule A166, this is actually a trastuzumab DAR2 conjugates, where they're using this novel auristatin derivative here, but again, leveraging a really stable connection to the antibody. And at a high level, they saw a great response in the clinic to this molecule, but again, very significant payload-mediated tox with this peripheral neuropathy, but again, saw ocular toxicity, and they actually saw that in all of the patients that they dosed with this drug. And so kind of the take-home here is this robust efficacy is really something that you can attain with more stable molecules, but now you're bringing in more intact ADC into tissues where you weren't driving that much ADC and payload before, and that's leading to new and potentially more severe tox down the road. And so turning back to where I had started and thinking about this target-independent uptake, this off-target, off-tumor toxicity, right? We want to think about how the ADC is actually getting into these cells, right? I just spent the last couple of slides telling you about ways that people have tried to mitigate these unstable linkers and releasing payload that's then getting into cells and causing tox. And while that does seem like that's helping in some respect, the ADC itself is now getting into cells and tissues and causing tox that way. And so if we go back and think about some of the ways that the ADC could be getting into these cells, again, we have target-mediated effect up here, whether it's the target antigen, Fc gamma, C-type lectin receptors, and other receptors that are specific to bringing antibodies into cells are ways that the molecule can be getting inside. But you also have non-specific endocytosis. And this is, again, where you're just getting the ADC itself being endocytosed in a non-targeted way into cells. Both of these mechanisms then can lead to the endolysosomal compartment where you can get FcRn-mediated recycling, which can then push back out your intact ADC. And so you can imagine there's a lot of different levers to pull on here, which I don't have time to go through. But one I wanted to spend a little bit more time here at the end talking about is this nonspecific endocytosis. And one of the things that is thought to drive that is actually lipophilicity for ADCs. And so you've heard a couple of times today that as we conjugate more and more linker payloads, greasy linker payloads onto these ADCs, we're actually increasing the lipophilicity of these molecules. And if you actually look at this via hit chromatography, where a longer retention time indicates a more lipophilic molecule, the naked antibody is here on the left. And as you go from DAR2, 4, 6, 8, you dramatically increase the hydrophobicity of these molecules. But importantly, the implication of this is actually you erode the PK profile for these molecules. And that's what's seen on the right here. So this is the exposure profile for the naked antibody, the line here on the top. Then you go to DAR2, DAR4, and DAR8. And as you increase that DAR, you can clearly see that erosion in your exposure. And so as a medicinal chemist and trying to correct this type of lipophilicity problems, one of the places where folks have tried to go is looking at polar groups to try to reduce this effect. And Seagen has actually spent quite a bit of time in looking at these types of molecules. I just pulled one of their publications here. In this case here, they were looking at stable DAR8 MMAE ADCs. And what they were looking at is specifically different lengths of PEG to try to see if they could reduce that lipophilicity of these molecules and potentially then improve the PK and tolerability. And so from the PK perspective here, when you look at PEG 0, 4, 8, and 12, at a 3 mpk rat PK dose, you can see a significant improvement as you increase the number of PEGs, i.e. reducing that lipophilicity is resulting in a much better PK profile, whereas at this PEG 8 and PEG 12, now you're actually starting to look like that parent MAB PK profile. But importantly, you don't just have an impact on PK, you actually have a significant impact on the tolerability. So this is a single dose rat tolerability experiment, 20 mpk. PEG 0 actually led to all animal mortality in less than a week. PEG 4, significant mortality. But then PEG 12 and PEG 8 actually dramatically improved the tox profile for these molecules. And if you dig into some of the data behind some of these experiments, particularly with regard to tissue payload concentrations, and I pulled two examples here, so one in the bone marrow, the other in the plasma. You can see for the PEG 0 as well as the PEG 4, this significant Cmax, very high Cmax for payload in these tissues compared to the longer PEG units, showing again that rapid clearance is leading to a lot of free payload in these tissues, which can be leading to that tox profile. And just to summarize some of the cytopenia data here for these compounds in particular, the impact on reticulocytes, you can see increasing that PEG, dramatically preserved those cell types. Similar for platelets, again, the longer PEG units were leading to significantly less platelet loss. But the data is not all positive. If you actually look at neutrophils, you still see a significant impact on the viability of those cells. And so while it's moving in the right direction with regard to improving the properties and the stability of these molecules, there's clearly some work to be done. So with that, just like to summarize, I hope I've convinced you with the limited time that I had that more stable linkers aren't necessarily the best way to solve some of this free payload toxicity. You can get improved efficacy in some cases, but it actually can lead to either similar or different tox when you actually take this to the clinic. But also too, this ADC payload toxicity is really a multifaceted beast, and a lot of the improvements that we have to do are going to have to be across the molecule itself in order to resolve some of these effects. Again, I showed you one example with regard to lipophilicity, but there's a lot of things that can be considered, particularly with regard to antibody engineering, which I hadn't talked about at all today, but is a really appealing direction to go in trying to solve some of these toxicity effects. But then also too, harkening back to the trastuzumab DXd data, the payload choice actually can play a significant role in the tolerability and ultimately the efficacy profile. And so with that, I'd like to thank you for your attention and happy to take any questions.
Thank you so much. You're probably the only guy talking about the preclinical toxicology profiling. I think it's very significant and important when we try to have a balancing between the efficacy and toxicity when we prescribe, the ADC. But one thing that missing is that we seldom talk about the ADC activated the complement system. It had, both direction. It can enhance the, cytotoxicity, but at the same time created a adverse event. Can you, comment briefly on the complement system, in the context of the ADC? And also, in clinic, we see a lot of, well, not a lot, but some proportion of ILD, interleukin lung disease, particularly for the TDXT. It ranges from 16 to, I would say 10% to 20%, or maybe 26% of interstitial lung disease.
Yeah. So let me just, for your first question, you were asking about complement? Is that what I heard? Yeah. So I think there's two schools of thought on that one. One, I think there are folks that think the FC-mediated functions are actually really important in helping to drive ADC efficacy. I think there's another camp of folks, of which I think I am a part of, that want to holistically drive our PD effect based on the payload itself. And so if that's the case, then you really want to focus on moving away from those immune-mediated toxicity events to try to give us one property to optimize and focus exclusively on that. Sorry, your second question?
Oh, the other one is the kappa, the complement activation related, pseudoallergy. That's why you mentioned about this rash.
Yeah. Well, so for the rash, that was specific for the Nectin-4 ADC and MMAE. Okay. Okay.
Hello. I was interested in trying to understand better, like at the end, you have some graphs about like some of the immune cells. So my question would be like, what are the current ways that you can evaluate the payload or like the delivery or toxicity more related to the immune response, or like what are the maybe like current research on that that can help us to understand better also the mechanism of the cells, or like how to deliver more efficiently?
Yeah, so I'm not an immunologist, so I think I probably have to defer the question around what particular assays can be used to leverage or to mitigate that tox. So I think one of the things that has been a particular problem for a lot of these models, and actually Pat LaRusso gave a talk on this at ASCO a couple of weeks ago, is actually some of the translatability for some of these preclinical models compared to what's observed in the clinic. And so I think in trying to think about what models or systems to use to try to assess that, I think the big unanswered question actually has to be the translatability.
Hi. Thanks.
Very quick question.
So you had the Trodelvy example and the KIG, the other example, where the only difference is how it's linked to the antibody.
Yeah.
And that carbamate is usually thought to be the weak point. So how do you explain the difference? Is it because it retromichaels to something else and then the linker falls off, or?
Yeah, so there's probably not a simple answer for that, but I think one of the ways you could answer that at a high level is that because the ADC is more stable, it does, in a targeted sense, deliver more payload to the tumor. And I think we heard a couple of times earlier, the more you can deliver in a targeted way your payload, the more efficacy you can see. And that could be partially what's explaining that. You just have more intact ADC and thus more payload in the tumor.
But it's more stable.
Correct. Yeah. Yeah. It's on the spectrum of stability, right? It's not as unstable as Trodelvy, where you have 2 points of instability. We've taken out one of those points, but still kept the other one.
Okay, great.
Okay.